Hello and welcome to the Illuminating Immunity Virtual Summit. I am your host, Dr. Joe Doduro, and my guest is Professor Dr. Paul Chazot. Paul is an associate professor of the Department of Bioscience at Durham University in the UK. He's very, very interested in research that focuses on identification of characteristics of new novel target drugs for the treatment of major and acute chronic CNS pathologies. And his background in the molecular aspect, as far as the European Histamine Research Society and the British Pharmacological Society, allows him to give us a great insight into this light and photobiomodulation therapy versus COVID-19. Thank you so much for attending today, Paul.
Hi Joe. Yeah, all good. Hi.
You said you've been putting together a review for a class or for research, or what? How did this kind of come together?
Okay, so as you said, I'm a neuroscientist, so you might think, okay, what does a neuroscientist know about COVID-19? Not a lot, I must admit. I'm not an expert, I'm not a virologist, but saying that, I've spent a lot of time working on photomodulation in the brain, and during that time, I've been mostly interested in working out mechanisms. So mechanism of action of these light therapies is what I do. And along the way, we come out with different mechanisms that we've found, and many other groups have found that certain wavelengths elicit in the brain, but probably will expand to all over the body. So, the understanding of the mechanisms helps then to think about, okay, COVID-19 is a virus, and how does a virus act? Where does a virus act, and what aspects of the viral infection can we look at with light therapy based on the mechanisms we know that the light therapy elicits in the body?
Perfect.
So that's where I'm coming from, basically. So it's what I've put together, and I'm writing a review, as you say, for British Pharmacological Society, British Journal of Pharmacology. At the moment, they've invited me to write a review on this to discuss the potential mechanisms that could be helpful to combating COVID-19, using the understanding of the mechanisms that many of us have been working on for many years for the different wavelengths. So that's where I'm coming from for this particular presentation.
Excellent. This is what we need. Well, let's go to the slides.
Okay. So, yeah.
Very good. Light at the end of the tunnel with a question mark. Tell me.
Okay. So you might recognize this image to the left of that title. That's our friend COVID-19. Many pictures of this you've seen in the press and in the media, in the scientific papers. So what I'm talking about today is the possibility of using photobiomodulation as a means of therapy for combating this present danger to life and limb within humanity across the globe, namely the COVID-19 virus. So this is the scope of my talk. So I've also quoted my friend Dylan Thomas. Again, we work on photobiomodulation and light. So this quote I always like to use in presentations for when I talk about the brain and when I talk about other aspects of photobiomodulation. Okay, so if we go to the next slide, I'll start to talk about the project. So this slide shows you what many of you, if you work in this area, photobiomodulation, know is the range of wavelengths that we have with light, ranging from gamma rays, X-rays, right down into visible range and then into the infrared range. And it's obviously the infrared range that most of us are working in, although there is some very exciting work also in the visible range of light. And this is what we're going to be talking about. So we'll be looking at photobiomodulation in this range, and I'll be focusing at the far infrared range. So working on a lot on this wavelength around about 1068, 1070, 1072 nanometers. And we've been working on this in the brain for many years. But also you have to bear in mind that there are other wavelengths that potentially are useful in photobiomodulation in therapy in terms of COVID-19 as well, but I won't be dwelling on those today. Other people will be talking about that in this conference, I'm sure. Okay, next slide. Right, okay, so again, this is a sort of something I've taken, again, from the grand literature out there at the moment. So this was published a few weeks ago, showing the COVID-19 and the basic elements of the COVID-19 and how it affects in the lungs. So we have the sort of the normal situation, the top right.
Sorry, there you go. Go ahead.
Okay. Yep. So on this slide we have a depiction of most of the things we know about COVID-19, the sort of normal lung structure, and then as the disease develops, gets worse over time, and it shows you some of the key things that happen within the infection. I won't dwell on this slide too much, but it's there for information later. One thing it doesn't actually have on this slide, because it shows you how things are moving so fast, is actually the very recent, in the last week or so, information and publications that have come out that suggest that actually thrombosis and clotting is a very important part of the pathology within COVID-19 infection. In fact, maybe it's one of the major reasons people die from the COVID-19 infection, and that wasn't really appreciated until relatively recently. An Italian study was published at the end of last week, looking at post-mortem individuals that had COVID-19 infection, and shown that 100% of the patients they looked at, or the individuals they looked at, actually had a very serious thrombotic pathology, and probably accounted for their ultimate death from COVID-19. So it's clearly an important part of the disease, and we'll talk about that later in my presentation.
Very good. We did get to cover that in another talk. We went into that in quite
excellent. Excellent. Yeah. Okay. Right. So again, this is another sort of introductory slide, showing again some of the pathologies and it's this sort of pictorial representation of what's happening in COVID-19. It represents what's the testing, it's representing some of the pathologies again, and what happens from the viral entry or attachment to the host cell and the infection, and then the consequent effects on the immune response. So the immune storm, which I'll talk again about later on, relating to photobiomodulation, and its effect ultimately on the lung pathology. So I'll move on rapidly to the next slide. So what I'm going to be focusing on today is really to discuss the rationale for using photobiomodulation treatment for COVID-19. So there's no data with the virus or that I'm going to show you. All I'm going to do is show you the sort of pharmacological rationale, because that's what I do. I'm a pharmacologist, and I like to have a good rationale before we start using a treatment. So I'm going to discuss some of the evidence for that rationale. So we're going to start at the beginning. So we're talking about the COVID-19 virus, the so-called SARS-CoV-2 virus. And before it can do any damage, it needs to actually enter a host cell. And we know that COVID-19 virus, the virus itself, attaches to the outside of the host cell, and most focus is on the lungs and the pulmonary cells, but actually the COVID-19 virus has a capability of binding to lots of different cell types in the body. And we know a lot about how that happens, and it seems to bind to specific proteins, this ACE2 protein on the surface, with the help of other proteins and other to facilitate its entry into the host cell. As I said, most of the work and most of the evidence suggests that the pulmonary lung cell is one of the most sensitive to this action, and probably the reason why the lungs are the most affected is the ease of entry of the virus into the body through the nasal passages and through the lungs. Whereas if the virus entered through the gastrointestinal system, it probably would get inactivated in the stomach, etc. So it wouldn't actually ultimately hit targets further down into the body. So there is a reason for it, but there is still a capability of the SARS virus to actually attach and infect other tissues in the body. And similarly, also there's possibilities of entering the brain, and there's evidence growing that the SARS virus is getting into the brain and causing problems within the brain. If you've got a leaky blood-brain barrier, for example, if you're elderly, you've got more access potentially through the nasal passages to enter the body and into the brain and cause problems within the brain. And there's, again, quite a bit of evidence of brain pathologies and brain symptoms following COVID-19 infection. Anyway, so the first point that we need to think about in terms of the rationale is the actual attachment of the virus to the cell. If it can't attach to the cell, it can't enter the cell. So that attachment point is an obvious drug target, an obvious target for thinking about, can we stop the virus actually entering the cell? As yet, there's no evidence out there that for changes in those sort of key proteins, the ACE2 proteins and the furin proteins, which again I'll mention in the next slide, that aid the viral entry. There's no evidence that photobiomodulation modulates those proteins, but those are experiments actually to be done. And as far as I know in the literature, there's no evidence. I may be proved wrong. Some of my colleagues may have investigated that in the last few months. Anyway, so we got no evidence at that point for the entry point that photobiomodulation has an effect. Okay, so we move to the next slide. So as I said, you need specific proteins on the surface of a host cell for that virus to attach. And the reason why this particular virus seems to be highly infectious, maybe compared to its predecessor, the SARS and the MERS virus, is that it also has other proteins like the furin proteins that seem to aid and facilitate the entry of the virus into the cell. So, and also allows for very strong sticking of the virus to the outside of the cell, whereas in the SARS, the SARS virus wasn't such a high affinity interaction with the host outside surface. So there are reasons why the COVID-19 seems to be highly infectious. Once it's attached, it stays there and gives it much more chance of entering the host cell. Whereas in the SARS virus, if it's low affinity, it's more chance of it actually then washing away, if you like, from the outside surface. Okay, so as I said, there's no evidence, as far as I know, of photobiomodulation modulating any of these sort of key proteins in that process. But we move on to the next slide. However, in terms of, okay, so assuming the virus enters the cell, and we're talking about, again, with a pulmonary cell, one of the things that actually that the photobiomodulation does do, and we know it does from a number of wavelengths, including our PET wavelength, i.e. 1068, 1017 nanometers, is that you get an induction of inducible nitric oxide synthase in many cell types, including the brain. And obviously in the brain it has its importance and has its uses in the brain. But it does, that fact is very important. And one of the key things about that action and that induction of nitric oxide is that that is an antiviral mechanism, if you like. It's well known nitric oxide is a very potent inhibitor of the viral replication process in many different ways. So it is an inhibitor. If you've got a rise in internal nitric oxide, you've got a potential antiviral mechanism there. And we know, like I said, 1068, we see an upregulation of inducible nitric oxide synthase fourfold in lymphocytes when we did our initial studies back in 2005, which was the first real description of that process, and many other people have subsequently shown that induction of nitric oxide synthase enzyme activity and consequent nitric oxide in the brain, and it's one of the key mechanisms in its brain action. And I'll talk about nitric oxide in other positive effects on nitric oxide later on. So the first thing to bear in mind is, photobiomodulation has a potential of inducing nitric oxide. And that's not just 1068. We know the near-infrared 670 wavelengths do similar things as well. So that, again, that could be an advantage of that type of photobiomodulation. Okay, so in terms of viral, as an antiviral, it has its potential already. Okay, so we've moved to the next slide. Okay, so this is just the old data. Back in, like I said, in 2005 and 6, we showed this upregulation of nitric oxide synthase in lymphocytes. In this study, we were looking at the effects of cytoprotection in human lymphocytes of 1068, or in this case 1072, so basically the same wavelength. Specific effects and most potent effects of that wavelength on these two processes, protecting against UVA damage in the lymphocytes, and at the same time simultaneously inducing nitric oxide synthase production. And this is an immunoblot showing up on the first lane is the control. Second lane is the light treated lane, where you see just by eye you can see the upregulation increase in protein expression. That's the nitric oxide synthase. And actually the third lane is a wavelength that it's not one of our favorite wavelengths, although again a lot of my colleagues work in this wavelength, where we actually saw no effect of the 880 nanometer treatment. And that again was our initial experiment. That was our first paper we published showing, and that's why, one of the reasons why we've stuck with this wavelength, the far infrared wavelength 1070, 1068 for subsequent studies in the brain. And obviously when you induce nitric oxide synthase, you induce, the presumption is you're producing higher levels of nitric oxide in your cell. In this case it was a lymphocyte, and we know also it can produce it in nerve cells, in neurons, and the assumption is in macrophages you could potentially induce it in many other cell types. So that's the basis for the nitric oxide as one of the key mechanisms. Yeah. Okay. And so the next slide, please. Yeah. So yeah, and this is in words. Again, this is sort of repeating what I've said, but I just had it on the slide for people's interest. If they, they'll have this available as the PDF after my talk, so they can read through the various effects of nitric oxide as an antiviral. So it's an antiviral, but that process is also part of the innate immune system, and that's very important actually, and we'll talk about that later on as another key effect of the photobiomodulation with the 1068 nanometers is this induction of the innate immune system. And part of that process is this inhibition of replication of viruses, including, as I've highlighted, the coronavirus class of viruses. And it's actually quite a wide range of effects on different herpes virus, for example. And in fact, that actually accounts for our initial, my colleague Gordon Dougal's initial interest in this treatment, because he actually developed it as an antiviral treatment for herpes virus, so cold sores. So it's an NHS approved treatment for treating cold sores, which is based on the sort of herpes based virus. So part of that mechanism.
Just a note, so that when they say that photobiomodulation or light therapy has an antiviral effect,
yeah,
it's because it's affecting this, and one mechanism is it's affecting a potent nitric oxide release.
Yeah, it's potent nitric oxide release, and that has but as we'll see later on, some other effects as well. But one of its key things is it seems to, it's well known as an inhibitory action on replication of viruses. So we can't, at the moment, we've got no data that it stops it entering the cell, but this effect of raising nitric oxide would be predicted to inhibit replication of the virus once it's in. And if you inhibit replication, then you're obviously halfway to stopping the virus, actually, stopping it at source to do any damage to the cell and to the body. Okay, so that's the first thing. That's the really important thing, actually. Okay, so the second thing. Okay, so assuming we're not treating the individuals particularly well, they come in with the virus infection. The next stage, once the virus is taken effect and has not been sort of stopped from replicating and from causing problems, the next big effect is what was called the immune storm, which is quite a dramatic description, and it's all over the press. Obviously, that they like to use this term, but it's a pretty good way of describing what happens, where you get this mass immune response to the virus. Once it's reached a certain level, I have to emphasize that. So once it's reached a certain level, you get this immune storm. But if you can stop it getting to that point, i.e. have a treatment that raises the nitric oxide or actually induces this innate immune system, so you stimulate the natural immune system to combat the virus before it then hits the point where you get the immune storm. So that's just to bear that in mind for later on, because that's quite important and is very relevant to photobiomodulation. Okay, so the immune storm is this, later in the development of the viral infection, you get this massive release of multitudes of cytokines, but very active cytokines into the system, which then obviously has mass consequences on the lungs and all the tissues around where those cytokines are released. So you start to get damage, you start to get more, sort of a knock-on effect on cell death. And the other big thing that happens after that is you also get potential effects on the vasculature, on the systemic blood system, and it also induces this clotting, this thrombotic pathology. So the immune storm comes before the thrombosis, generally speaking. So that's the stage of it. So you get the viral infection, you then get the storm, then you get the thrombotic. So these are the three main stages that we're talking about.
Right.
Okay, so if we move to the next slide, and then this will show you some data. Or actually, yes, this first of all, this is an interesting slide, because one of the effects that we found when we've used our wavelength, the 1068, in the brain, and we found is induction of a whole range of different heat shock proteins. One of those heat shock proteins is heat shock 70, protein 70, and again, there's some data which is related to our brain work, where we show induction when we treat animals in the brain, and the brains of animals are analysed. You see an upregulation of this heat shock protein 70, and heat shock protein 70 in the brain and in other cells is very important in protecting brain cells from cytotoxic stress. It acts in the mitochondria. It improves oxidative viability of the mitochondria, etc., etc. It's very important in proteinopathy phases of cell pathology. So in the brain, and in brain disease, we've seen this upregulation, which we think is very important for coping with the pathology you find in Alzheimer's disease, for example. Anyway, the interesting thing is heat shock protein 70 also has a very important role in the story in macrophages. Heat shock protein 70, and because actually going back, taking a step back, one of the key things you see in the COVID-19 infection is a rise in temperature. So you get this pyresis. So one of the symptoms that you're asked to look for in individuals is a rise in temperature, and that has a consequence on heat shock protein. By its name, you can see heat shock protein. Actually, that rise in temperature induces upregulation of heat shock protein 70. And that is actually, is a protective mechanism in the body. And this heat shock protein 70 has some interesting effects also on viral infection itself. So the virus causes the problems, the rise in temperature. It causes heat shock protein to increase as a sort of a protective mechanism. One of the ways that heat shock protein 70 protects is to actually be involved in the viral infection itself. So it's a sort of a negative control over the viral infection. And it does modulate, and again, it induces this innate immune system to then combat the viral infection as it's going on. So this effect is interesting. This heat shock protein rise is interesting. And we see it also with the photobiomodulation. So this could be, again, is another reason why potentially that this method, the 1068, might be a useful strategy, because it naturally raises the heat shock protein 70 expression. Because one of the problems of raising the temperature, obviously you want to control that. So what you would do is take a drug to bring down your temperature, but that would actually stop the heat shock protein 70 being induced. So if you use the photobiomodulation simultaneously to raise the heat shock protein 70, you'll then get the effect, the beneficial effect, the antiviral effect, the inducing of the innate immune system to combat the viral infection, if you see what I mean.
Correct. And now we're seeing this data was in microglial, okay?
Yes.
But
this was in neurons, so it's in nervous tissue, yeah. I'm looking on the right. The right-hand side is in brain cells, okay?
Correct. But my question is it going to have the same effect in other tissues?
Well, the assumption is basically the photobiomodulation, whatever effects it has in neurons, will have effects in all cells, because its main target is the mitochondria, yeah, and mitochondria is every living cell in your body. So the initiation of all these effects of the photobiomodulation, i.e. the effect on mitochondria and then its effect on gene expression, will happen. The assumption is it will happen in all cells.
Correct.
Okay, so that's where the leap is. So you take that, we work in neurons, we work in glial cells, we work in other cells, so we work in lymphocytes, as I said, in the immune cells, and we have the same effect. We have similar effects in a lot of these effects of the photobiomodulation. Okay, now I have to put a little caveat in that, if you don't mind, if we go back to the previous slide, just to have a caveat. Okay, so when you look at the data with heat shock protein 70, it's very viral specific. Okay, so there's been published work looking at this heat shock protein 70 effect, and it seems like a rise in heat shock protein 70 in certain viral infections actually has the opposite effect. So it may not be, it's very viral specific, i.e., for example, in the Zika virus studies they've done looking at heat shock protein 70, they do get, they suggest that actually a rise in heat shock protein 70 is not a good idea. Okay? But the good news is when it's been done with a sort of a coronavirus type virus, it seems to have this effect, as I'm showing you in the diagram on the left, where it's inducing the innate immune system. So there's a bit of caveat there. Okay, it's not a general effect across all viruses, but there is a suggestion, and the data is obviously quite small, the suggestion that it would give you a beneficial effect in a coronavirus type system.
Gotcha.
Okay, so it's very viral specific, it seems. Okay, so the way it's working is inducing this innate immune system. Okay, partly. Okay, so this is the interesting bit in terms of the immune system. Okay, which is the sort of topic of this conference really, is that there's different phases of the immune response to the infection. Okay, so you've got the initial response, which is called the innate phase, then you've got an acute phase, and then you've got a chronic phase. Okay, so in the innate phase, what you're trying to do, because you detect the virus. I'm saying the cells are detecting the virus, the body's detecting the virus, so it triggers its innate system, i.e. releases a lot of immune cells, a lot of cytokines, a lot of things that want to actually kill the virus or stop the virus from working. So you want to actually stimulate the inflammatory system at this stage, in the early stage, okay? Then you get to a stage where, okay, you've perhaps got it under control, but then the virus is sort of coming back again, and then you cross over into a sort of an acute phase, and different cytokines again are affected in this acute phase, and different effects happen in this acute phase, and then you've got a chronic phase. So this happens over time. So when we're talking about the innate phase, that's maybe over the first few days of infection, okay? And the symptoms may not be as severe, so it's sort of the early stages, if you like. But if it then deteriorates, then it goes into the sort of the immune storm, and then you have the chronic phase where you have the pathological effects on the lungs. Okay, so in the serum of COVID-19 patients, different cytokines and different molecules have been looked at, and you get a initial immune response, the innate phase. You get this, and you get a mixture of different cytokines, including, I've highlighted some here, sort of interleukin-1 beta, for example, TNF alpha, etc. There's lots of different cytokines trying to combat the virus, the viral infection, and then that changes over time, obviously. Then you go into a phase where you have too much immune response, and then you get, the pathological consequences, which ultimately leads to serious symptoms and death. Okay, so that's very important to bear in mind, because the effects of the photobiomodulation of 1068 fortunately seems to follow this pattern. Okay, so if we go to the next slide. Yeah, go to the next slide. We have, okay, so again, I've sort of split it into two phases. You've got the early signs, the early stage, and then you've got the severe stage. So the severe stage is when we really have the sort of, the storm, the immune storm. But the early phase is when we're trying to combat the virus. So we need an inflammatory response to that. So that's the early phase of the innate immune response. Okay, so when you treat, in this case, these experiments were done in skin infections, okay? And what was found with the photobiomodulation is that you get a pro-inflammatory response for the first day or so, okay? So you do get this early phase pro-inflammatory innate immune response where you get cytokines released, listed here in the slide. You've got interleukin-1 beta, TNF alpha, interleukin 6, etc., that are released by this photobiomodulation effect. But then over time, you obviously don't want that to carry on if the disease starts to progress into the immune response gets worse and worse. If the viral load gets heavier and heavier, you get a bigger response. So what you want later, you want an anti-inflammatory effect, and that in fact is what you seem to get with this photobiomodulation, with this wavelength. You start to normalize after three or four days, and then you have a chronic phase of anti-inflammatory effects. So you get downregulation of the cytokines, you get downregulation, you normalize the situation. So this is almost a sort of ideal scenario for what you want to combat COVID. You want something that's going to stimulate the system to try and combat the viral infection, and then you want something later, if that wasn't sufficient, to actually reduce the inflammatory, the immune storm, and reduce the severe inflammatory responses.
So I think this is a great description. So thank you for this slide. So,
But I have to say, obviously, this is done on very few studies.
okay.
Yeah, and it's done in different tissue. But the assumption is that it would do very similar things in the lung.
So,
Okay, but nobody's obviously that's not been proven, and I have to put all those caveats in. But it's interesting that the photobiomodulation response with this particular wavelength seems to follow this pattern.
okay.
Some of the other wavelengths seem to just give you a anti-inflammatory effect on its own, which is fine. But to have a treatment that gives you both is the ideal scenario, because you actually want something to combat the virus at the beginning, and then obviously, if that's not enough, to then combat it towards the end.
So
Of the process.
my question is that this seems to be a step on the gas, then step on the brake.
Absolutely. Yeah. So if you go to the next slide, it shows you the data. Just
Okay.
go to-
But my question is,
yeah.
is it you turn on the light and you got the gas and then the brake, or do you have to continuously put the light on to get the gas and continuously get the brake? Where's the?
I think that's a good point. I think you still need to carry on the treatments, because obviously what I'm showing you here is it's multiple effects. It's not just, we've got this immune effect, but also, like I said, we've got the nitric oxide effect, which is also another, again, as we'll talk about later, has other very useful effects downstream.
Oh, yeah.
So in other words, you need to have the patient being treated over the period, because you want them to give the best chance to the patient to recover from the viral infection, recover from the immune response, and also potentially, if it gets through all that, to then stop it affecting the clotting phase, the thrombosis stage. So it's the beauty of this treatment. It seems to be able to potentially has the ability to stop it at all stages of the process. So I think, it won't be a one-hit wonder. I think, like with light treatment generally, with all our work we do, most of all, for all the guys that work on photobiomodulation, you need to do multiple short bursts of treatment to give you the maximum effect. It's not just a single hit.
Perfect. To crank this up. Thank you.
Yeah, that's my feeling. Again, in terms of this, obviously we haven't got data to back it up, but this is just an idea. I'm just taking you through the rationale of why it might work.
Gotcha.
Yeah. Okay. So if you go to the next slide, it just shows you again with some numbers. So this is the data from the skin studies. Again, it sort of highlights the time-dependent effects they're seeing. So the table on the left shows you again the baseline, and let's just give you a few examples of some of these cytokines, and also shows nitric oxide synthase again at the same time. So initially the baseline is relatively low, and then you get this induction in most of these markers short term, so up to say 24 hours. So that acute phase, that innate, so you got raised levels of TNF alpha, for example, interleukin 1 beta, interleukin 6. The nitric oxide starts kicking in, so that's anti-inflammatory as well as potentially antiviral. And then over time, if you look at 3 days and 5 days, you start to get a more stabilized situation. Certainly for the TNF alpha and the 1 beta, that's still kicking in, but you're getting a stabilization of some of these parameters. And again, it shows the nitric oxide increase lasts over that period. And if you look at the NF kappa B images on the bottom, again, in indication of inflammatory effects. So again, comparing the 10, 6, 72 versus the control, you get different effects at different time points. Again, you get reduced NF kappa B, say at day 3 later on in the process. So again, reduced inflammatory responses. So you get some induction early on, and then you get some stabilization later on in the process for many of these key cytokines. Okay, so that sort of summarizes a little bit about the immune system. So if you move to the next slide, again, there is data. Again, this is again data looking at the virus and potential effects of the nitric oxide synthase, again inhibiting the immune response in this case, as well as the antiviral effect. So you get effects on interleukin-6 and interferon gamma, also which are effects on pro-inflammatory, that early phase effects. And then obviously, as I said, over time we seem to get stabilization of these effects. So it's a combination of antiviral and a combination of anti-inflammatory.
Very good, very good.
Okay, so if you move to the next slide again, okay, so that's a bit about the inflammatory system. So we've talked about the viral entry, viral inhibition. We talked about the immune storm, immune responses. Now we're going to move to what sort of seems to have is the flavour of the month, flavour of the week. Now, this idea that actually thrombosis and clotting could be something we should have looked at right at the beginning, as the major, one of the major causes of death within patients in intensive care. Like I said, this Italian study has suggested this strongly when they've started looking at post-mortem individuals, showing that 50 out of 50 patients they first looked at basically had clots in their thrombosis in their system, basically. So the immune storm elicits this downstream effect, is eliciting this thrombosis, this increase in aggregation of platelets and red blood cells in the pulmonary system, pulmonary blood system, basically. So if we move to the next slide, we can have a look at what's happening. Again, this is quite a nice diagram to show you what's happening here. So I mean, it's got the influenza virus as an example, but you can replace that with a COVID virus, potentially what's happening here. So you've got this cytokine storm leading to a thrombotic situation within the blood system. So this intimate connection between the immune system and the bloodstream and the blood system, and this idea that this increase in thrombosis, increase in clotting, ultimately is what causes death in many of these patients. So it's a combination of death from the immune storm, I guess, and together with the thrombosis are probably the two main causes of the deaths within COVID-19, and probably accounts for why people that were put on ventilators, individuals that got on ventilators, 60% of them actually didn't make it. Because ventilation, as I know personally, I've been ventilated a few times when I was in the ICU, is quite a high risk factor for thrombosis, because obviously you're stationary and you're on the ventilator. There's a high level of risk of thrombosis in that situation, and it's more of a risk on a ventilator than it is on a CPAP machine, for example, where you're just getting a device delivering you the oxygen. Whereas on an intubator, you've got much higher risk of clotting. So that's probably why a lot high percentage, unfortunately, died when they were put on the ventilator. If it could have actually just gone on the CPAP and been given an anti-clotting agent, maybe you would have had a higher survival rate.
And we sort of have that to deliver more oxygen and to create less viscous blood, less viscous blood, so that we have the ability to do that with our therapies.
Yeah, we do, in fact. And I think, talking to my colleagues in intensive care, that they are starting to look at that now. Well, they've been doing it for a while now, basically. I mean, they've noticed themselves on the front line that, these sorts of things are happening. It was only formally sort of reported last week, really. But yeah, so if you have an ability, again, to reduce that thrombosis, then you've got, again, another arm to to your therapeutic strategy. And again, looking at the literature, if you look at the literature, there is already a study out there, and this is the one I'm showing you here, using far infrared therapy treatment in quite a good journal, showing, and I'm showing you just some of the data. This data relates to the thrombotic idea. So that what they've done is they've shown treatment with the photobiomodulation treatment reduces collagen and ADP induced platelet aggregation. So that's shown in the slide. So the darker bars are the treated samples, and again that's over a period of time, up to 24 hours. Here you can see a clear reduction in this aggregation induced by collagen and ADP. These are classic ways of inducing aggregation in blood vessels. Yeah. So the photobiomodulation seems to have this anti-aggregation capability. And also, if you go to the next slide, importantly, because this is something you need to know when you're in intensive care, okay, when you're using a...
One second, I just want to do a shout out to my collaborator, Thomas Walsky, the third author on this. And
okay, yeah, good.
And he has a recent paper a couple months ago on the anti-inflammatory responses of the light that he's seen, and that paper is going to be reviewed in our program. So he's currently we've been working together for about a year and a half now, and these are the sort of things that he's talking about because he's the man when it comes to the blood.
So you can combine the two, right? Yeah. So in terms of this idea, you can put the two together basically. So it's two positive effects to combat the two key elements probably of what's killing people basically. So importantly, if you go to the next slide, yes, one of the things, again, when you talk to guys in intensive care, the docs and everything, the guys working there, when you're dealing with a thrombolytic treatment, you've got to be very careful. Because you want something that's going to reduce the clotting, but you don't want to get to a point where you get bleeds, so hemorrhagic situations. So this was an interesting study, again, I picked up in the literature, which importantly has compared the effect of TPA, which is a sort of classic thrombolytic, for stroke situations. What they've done is that it shows that because there's a possibility with TPA that you do get hemorrhagic problems, obviously. And again, you have to distinguish when somebody with a stroke, you have to distinguish between a stroke or a hemorrhagic stroke. Otherwise, you kill people. Basically, if you give a TPA to a hemorrhagic stroke, that's not good news. So it is an important issue. So the important thing from this study, from this graph, if you see, so this is the level of hemorrhage with TPA, which is always a risk when you're given TPA. But when you combine it with the treatment, with a transcranial, in this case a near infrared, so I assume it's sort of more in the 600 range, it doesn't exacerbate that, so it doesn't cause extra problems on top of the TPA. So you can potentially use this in combination with a thrombolytic. That's which is quite an important thing. So so the previous study showed that it's anti-thrombosis, anti-clotting, but importantly, if it's combined with other thrombolytics, it doesn't cause more risk of a hemorrhagic a stroke or hemorrhagic problem. That's a good combination. It's always, you have to think, you have to think of both sides of the equation all the time. So you make sure that you don't get drug interactions, which again was a worry when you start to work with thrombolytics. So that's good news. So, and again, it's further evidence, and in fact that's evidence with wavelengths that we're using. That you've got an anti-thrombolytic, anti-thrombus, anti-clotting effect, which, as I said, could be one of the key reasons why people are dying from COVID. Okay, so just an extra little bit of it to go along with that. Obviously, when you've got clots in the bloodstream. Again, another useful property of the nitric oxide induction is increased blood flow and vasodilation. Again, you do get vasodilation in the early phase, in the acute phase, because you need to get immune cells to the point of infection quickly. So blood flow is a good thing initially. And again, the nitric oxide helps with that, but also it helps generally to relieve the possibilities of clots. So the combination of a anti-clotting and good blood flow, and if you go to the next slide, well, the next slide again shows you the effects that we see with nitric oxide. You get induction with the nitric oxide. And if you go to the next slide, this has effects on the vasodilation, very well known effects of nitric oxide on vasodilation. I think it might have won the Nobel Prize a few years ago, that observation basically. So if you then go to the next slide, this is some data again with some infrared light wavelength irradiation. Again, is very well known in the brain studies as well, and this was done in the brain, I think. No, actually it wasn't. It was done elsewhere in the body, but it's the same principle. Rise in nitric oxide with the photobiomodulation, you get increased blood flow, and there's some nice images here showing increased blood flow within the system. I think this is actually a arteriosclerosis potentially individual.
Showing right here,
so again,
this increase in pre and post right there in the circle, and in the lower one, I'm going to wait.
Yeah, that's right.
Circle. It shows the vasodilation.
Yeah.
And this looks like the retina.
Vasodilation.
This looks like a retina.
It does actually. Yeah. It does look like the retina actually. Yeah.
I think that's good photo.
Yeah, you could be right, but it highlights your question actually, that okay, you show it in one cell or one structure, and I'm saying to you, the effects of photobiomodulation and the mechanisms are actually across the whole piece, if you like. All different cell types, all different parts of the body, be it in the retina. Now we're starting to do some work now in the retina. Actually, we're working on retinopathies with photobiomodulation, actually showing nice effects on retinal function and protection, neuroprotection, and glial protection in the retina. So yeah, it crosses a whole range of different tissues, a range of different cell types, because ultimately the mechanisms go across all cells.
Right.
Okay, so if you go to the next one, I think we're getting near the end now. Okay, so the final thing, again, the final bonus, which is actually what everybody knows really, the effects of photobiomodulation is to actually protect cells from oxidative stress and other stresses. So this is already known, and it's in the literature. And again, if you talk, this is what we study all the time in the brain, and trying to look at neuroprotective mechanisms. And again, I need to emphasize these cytoprotective mechanisms. All these mechanisms across all cells. So the idea is here, as well as treating the virus, combating the virus, antiviral, or anti-immune, or anti-clotting, it also, at the same time, theoretically, should be protecting the host cells from all this as well. So it's, you get this effect, known effect on nitric oxide, reactive oxygen species, ATP, and mitochondrial oxidative stress protection, etc., etc. So all these, you can imagine, again, in the pulmonary cells is going on as well at the same time to protect those cells and any other cells that the virus infiltrates, immune cells. We've already, our first study back in 2005 was on lymphocytes, human lymphocytes, again, the immune cells themselves. Some of the important immune cells like lymphocytes, which you need to combat infection, are protected as well. So it's that's the final, if you like, point, if you like, that it protects the host cells from the viral infection at the same time. Okay, so if you go to the next slide, again, this is sort of more If you go to the next slide. Okay, so again, so that you can imagine, again, this is just an example of a picture of the pulmonary cells, the epithelial cells that are affected by the virus. Again, to protect them, you've got mitochondria there. You're protecting against oxidative stress with the well-known effects of photobiomodulation, both at the high end and as well as the near infrared range is well-known effects on mitochondrial viability, etc. And again, the effects on macrophage function as well, protecting macrophages when they need to combat the infections that you're having. Because the other important thing to bear in mind is that, and again, another common cause of death within this COVID-19 situation is secondary bacterial pneumonia or viral pneumonia. So you sort of survive the COVID, and then a lot of people are dying also from the secondary pneumonias. So again, you need to think of that as well. I haven't put it in my talk, but obviously, the photobiomodulation can also protect against bacterial infections as well through similar mechanisms I've discussed with the virus, in terms of the virus. And in fact, there's more data on antibacterial effects of photobiomodulation than viral effects. Okay, so again, you've got you've got that another layer of protection, potential antibiotic effects of the photobiomodulation to make sure that the individual survives through our post-COVID, because obviously once you're post-COVID, you are very weak and very and you are very prone to secondary infection. Okay, so the final, we're getting there. Okay, this is just this is just a summary of some of our mitochondrial effects, our positive mitochondrial effects on complex on various complexes 3 and 4 in the mitochondrial exchange. Again,
basically, just to summarize a little bit, you're saying that if we can get the photobiomodulation into the lung cells or near the lung cells, they're going to have stronger ability to survive. They'll survive. They will be able to have more energy, more ATP, more energy to stay alive from as they're going through the process. So that's as
they're going through this stressful stage.
Correct. That's like a super bonus at that. Like while we're combating everything else, like you said, the autoimmune aspect, the inflammation aspect, the clotting aspect, we still can protect the host cell, and in this case, the epithelial cell or the brain cell, from running out of juice and just dying, because that's what happens when you die. You run out of energy, right?
Yeah, in essence, because I mean, obviously the COVID is really draining the cells, basically, in essence. So they are under severe oxidative stress. So if you've got a system that you're treating versus the virus, and at the same time it's also keeping the host cells as healthy as they can be. So you maintain as many of the host cells as you can during the whole process. So again, there's clear evidence for that side of things is a definite a tick. As I've shown in a lot of my slides, I've put a lot of ticks in the process, and I'll summarize the ticks in my last slide in terms of the effect of photobiomodulation. Just to finish off the effects of 1068. This is a study we've not published yet. This shows you the potential health benefits of this treatment using a classic aging model, the C. elegans worm, showing an extension of lifespan, showing that it does maintain cells for longer, and it maintains life for longer, if you like, at the cellular level up to the whole animal level. In this case, we showed this was a HSP70 mediated effect, potentially. If you remember, the treatment, our wavelength induces HSP70, which, as I've shown you in the beginning, has some potential beneficial effects in terms of COVID-19, antiviral COVID-19 effects. And this again is just another bit of evidence that HSP70 is important in the photobiomodulation of this particular wavelength that actually allows a worm to survive for longer, over 16% longer lifespan. Okay, so if you just click for the next, so that just shows you if you do it in a worm that doesn't have HSP70, you don't have the effect. So it's a HSP70 mediated effect. Anyway, this is the last slide. So this slide is designed to basically show you where we are in terms of the evidence. Well, for my idea that the 1068 could be the sort of ideal wavelength to try. That's not to say the 670 and the 850 may be suitable wavelengths for various different reasons. The reason I'm choosing this wavelength is it seems to actually cross the whole spectrum of potential protection and antiviral effects. So you go from, we don't know, like I said, just to summarize at the beginning, the viral attachment as yet. Those are experiments to be done, which I'm hoping to plan to do in the near future to see whether it affects the expression of these key proteins that are involved in viral entry. So the soluble furin is an interesting target because this protein is involved in allowing the viral entry through interaction with ACE2, but actually the soluble form of furin is a new, it could be a good inhibitor, if you like, of viral entry. So if it's in the soluble form, this furin 2 protein, if it's released and raised, can actually act as an inhibitor. If it's in the membrane, it actually does the opposite. It helps with the entry of the virus into the cell. So that's why I've got a question mark against that, because at the moment I don't know any evidence that something might be presented in this conference about that. But as far as I know, there's no evidence that those proteins are affected by modulation. Anyway, so the INOS effect is clear with the wavelength, the 1068 nanometer wavelength. You get induction of INOS, increase in nitric oxide. So that has an antiviral effect, as I showed you, a little bit of evidence. It has vasodilatory effect, has anti-inflammatory effect. So it has a triple whammy, if you like, of effects. That's one of the main effects that we've seen with many wavelengths in the photobiomodulation field. This HSP70 effect, this antiviral effect of raising HSP70, is a possibility. I'll put a question mark at this stage because the caveat is it doesn't seem to be potentially across all viruses, but it may be useful for the coronavirus. There's a little bit of evidence to support that this may be useful effect. Okay, so the innate immune system, as I've shown you, yes, that's induced with this wavelength, at least in the skin, but the assumption is that would potentially happen in the lungs as well. And you get this time-dependent pro-inflammatory and then anti-inflammatory effect, which again could be the ideal way of combating a virus like coronavirus. That you have to reduce its infection in the first place, and then whatever's left you combat with anti-inflammatory effects. Then you've got the anti-thrombotic effects, which I've shown you. Again, there's evidence already for that potential. Increased blood flow as well, obviously, which helps with the pro-inflammatory, the initial innate immune system to work, but also ultimately to stop any thrombotic issues. And finally, the host cell protection mechanisms, which are well established in terms of maintaining metabolic activity of the host cell, either in this case the lungs, pulmonary cells, and mitochondrial viability. So that summarizes where we are as far as I can see. And I'll be interested, obviously, to see if other people have some other ideas and they have other data that would fit, the question marks mostly in this process. And it would be good, obviously, to see if we can get it tested.
Yes, and I think that's one of the things that you're working on in the future is you're going to be one of the labs that's going to be doing it at a cellular level and maybe even the human level, right?
Well, that's the plan. That's what we're trying to establish. Obviously, we've got to, obviously get approval to do that in humans. It'd be good to get to that stage fairly as quickly as we can. And obviously, it's a treatment that is FDA approved for the brain and for other uses. And Marvin will explain, the methods that we can use. That they're already good to go in terms of strategies of how to deliver this light, obviously, to the lungs. Again, penetration of this particular wavelength we know is extremely good, so it will certainly get to the target, be it through the nasal passage or through across the lungs. So in terms of delivery of the light, it shouldn't be an issue at all.
Right.
But obviously, we need to demonstrate some efficacy against the virus would be good. And again, I'm in discussions with labs that can do viral COVID work, basically, to establish whether we get evidence for antiviral effects in the first instance, but ultimately, yes, obviously, to be tested in humans.
And if we look at your diagram on the other side before we end here, you put a little sunlight, you put light, and you put it in the areas that it really does affect, the microvessels, the inflammation, the cytokine storm, and in the host area. So like even in, say, heart failure or things like that,
exactly.
We're going to have the host cells. So you covered a lot of those in that diagram too. Thank you for that. Yeah,
that's right. I mean, those are the ones that we have, more evidence for, if you like. I mean, again, the evidence is, it's quite light. I have to be
correct.
But again, it's early days, but there are certainly some quite, if you put the whole story together, it's actually quite compelling.
Correct.
Potentially for its use, but obviously, we've got to get it tested as soon as we can.
And I say that it's only the people who know the literature that can tell the story, and you did a wonderful job. So I say thank you.
Okay, good.
And keep up the good work. Maybe we'll circle back in a month or so, because I think that we're all going to be working on this in the future for updates. Sure. And thank you for your work. I'll put it together, and your slides will be available at the end of the talk.
Yeah. So, okay. Yeah, if you make a PDF, that'd be great. Yeah.
Thanks, Paul. Thanks so much. Thank you for all your work.
Okay, all the best. Yeah. All right. Cheers. Ta-ra.
Cheers.